Hello, good afternoon, everyone.
Good afternoon, sir. Good
Good
afternoon.
afternoon, sir.
Yeah, great. How are you doing?
Yeah, fine, sir.
Okay, great. And
And
I'm
you?
getting ready for your examination. Um, this afternoon, I want to take you through cellular respiration. Um, and I'm going to flip through glycolysis, TCA cycle, and the electron transport chain. Okay, and I will start right away by asking that what do you think will happen if someone is not breathing? Okay, what happens when somebody stops breathing? Definitely that person will be dead. Will be dead because there will be no generation of energy to sustain the body. And so what cellular respiration is all about is that it's a set of reaction that takes place in an organism and is actually meant to convert biochemical energy to ATP. That ATP is very, very important in driving the activities in the body. And so if you look generally at the relationship between plants and animals, plants, which are referred to the autotrophs, they get sunlight and they utilize carbon dioxide and water to produce carbohydrates, which is taken by humans. The humans, we need oxygen, and then we use it to break down the carbohydrate that has been produced, and then we release carbon dioxide back to the plants, and then it goes on and on. And so humans or mans or animals generally are referred to as heterotroph. So generally in this course, we want to look at how those ATPs that is needed, how they are being produced. So that is what is of interest to us. Because like I told us, that the ATP referred to as adenosine triphosphate major currency of the body. So currency of the body because it helps to fuel activities that goes on in the body. So without it, there might be no activity. And if there is no activity, the living organism cannot survive. So the relationship, the interrelationship between the plants and the animal is very, very important because it is this activity that will eventually help to generate that ATP in the powerhouse. And most of this reaction that generates the ATP occurs in the mitochondria. And probably that's why mitochondria is referred to as the powerhouse of the cell. So it's referred to as the powerhouse of the cell because of the generation of the ATP, which is very, very important in the metabolic processes that occurs in the organism. So it's very, very key, very, very germane. So as plants, they utilize the carbon dioxide, the presence of water, and then when they do that, they produce carbohydrate, which is taken up by the animals and then many other microorganisms, bacteria, and then they use it in the presence of oxygen. It's being used to generate the currency, the energy currency of the cell. And that goes on to help the animals to actually drive the processes, several pathways that we have in the system. Several pathways that we have in the system. And so this goes on and on and on and on and on and on in the body system, because it's very, very key. Can you hear me?
Yes, sir.
Yes, sir.
Okay. And can you also see the screen?
Yes, sir.
Okay. So, like I said, I will take us through the various processes that are involved in the generation of the ATP, and then see other equivalents that also contribute to its generation, and then see the amount that is being generated at different pathways that we're going to look at. So like I said, looking at glycolysis, you're looking at the citric acid cycle, you're also looking at electron transport chain vis-a-vis oxidative phosphorylation, and their concomitant generation of energy. Great. So, like I told you that if you have the carbohydrate here, the oxygen being broken down into carbon dioxide and water, and 36 molecule of ATP. So this happens, this is the totality of what we're going to look at. So like I told you, we are going to look at glycolysis, which is the breakdown of glucose to pyruvate acid, the citric acid cycle, and the electron transport chain, all resulting into the generation of ATP. But of course, the electron transport chain is more in generation of the ATP. For the heterotrophs, which are the animals, we have the making of the energy. So the glucose is being broken down through oxidation. Okay, so we have it as hexagonic reaction. So we have CO2, H2O, and ATP, and here we have for the autotrophs, we have carbon dioxide, water, energy, and glucose and oxygen is being released. And this reaction is endergonic reaction. It's a reduction reaction. This one is oxidation. So, of course, we have different types of respiration, the aerobic and anaerobic respiration. The aerobic respiration is kind of respiration that requires oxygen for it to break down, while we have anaerobic respiration where oxygen is not needed. Okay, so when the glucose gets into the body, of course, you need the blood to take the glucose around. Then the glucose finds its way towards the cell. So when it comes inside the cell, you now have the process of breaking down the glucose that we are going to see through the process of glycolysis. So it is during this process of breaking down glucose to pyruvate that ATP is actually being produced, and this happens in the cytoplasm but the bulk generation of energy through the electron transport chain happens in the mitochondria. Okay, so let's look at it now. Glycolysis. I told you is the process of breaking down glucose to pyruvate and then with the concomitant generation of ATP. Now, glycolysis can be grouped into two phases. We have phase 1, and the name of that phase 1 is called end of investment or preparatory phase. That's the first, and that's the first phase we want to consider. So in this particular phase, glucose will break down to glucose 6-phosphate. Glucose 6-phosphate is broken down to fructose 6-phosphate. Fructose 6-phosphate is broken down to fructose 1,6-bisphosphate. And then this fructose 1,6-bisphosphate will be broken down into two interconvertible products. The first one is glyceraldehyde 3-phosphate, and we have dihydroxyl acetone phosphate. So glyceraldehyde 3-phosphate and hydroxyl acetone phosphate can interconvert. It's a reversible reaction. Okay, but it is one of these that is going to continue the journey into the second phase of glycolysis. So in the first step, we have glucose to glucose 6-phosphate. We are going to have an investment of ATP. We are going to put ATP going to ADP so that glucose will be converted to glucose 6-phosphate. And the name of the enzyme there is hexokinase. Then second step is a reversible reaction of moving to glucose 6-phosphate to fructose 6-phosphate, and the enzyme involved is phosphoglucoisomerase. Then the third one is converting fructose 6-phosphate to fructose 1,6-bisphosphate, and in this one you're also investing ATP and the presence of the enzyme phosphofructokinase 1. And then you have fructose 1,6-bisphosphate being broken down into glyceraldehyde 3-phosphate and dihydroxyl acetone phosphate. And the enzyme responsible for this breakdown is called aldolase. Okay, so having said that, it's also important for you to note the regulators of this particular pathway. Before then, we have hexokinase here. We also have another enzyme that can convert glucose to glucose 6-phosphate. The name of that enzyme is called glucokinase, but you can only find this in the liver and the pancreatic beta cells. That's where you can find the glucokinase. Outside that, it is hexokinase that's going to help you convert glucose to glucose 6-phosphate. Okay, so we have molecules that regulate these pathways at different points. Step 1, molecules that regulate it, we have insulin, we have glucose 6-phosphate, and then we have a glucagon. We have the glucagon. Okay, so insulin helps to activate the reaction. So when you have insulin present, okay, it helps to activate the reaction because it signals that, oh, there is enough glucose in the system. And of course, insulin helps to transport the glucose into the cell and subsequent conversion of glucose into glycogen. So that process, so when insulin is present, it can help to activate hexokinase to convert glucose to glucose 6-phosphate. However, when you have increase in concentration of glucose 6-phosphate, the first product, it is an inhibition to the conversion of glucose to glucose 6-phosphate. So when you have high concentration of glucose 6-phosphate, it's an indication that, oh, you have enough, and then the reaction should slow down. Okay, so we also have a particular situation where the molecule that is present affects the inhibition affect the inhibition of conversion of glucose 6-phosphate. Okay, so we have the glucagon as well, which has act as a negative control. Glucagon increases the blood sugar level. Okay, and what it does is that it helps to break down glucose that have been stored as glycogen. So in that point, it shows that, oh, probably you don't have enough glucose. So you are breaking down the glucose already stored as glucagon. So the glucagon itself serves as inhibitor to the pathway of converting glucose to glucose 6-phosphate. Another point of regulation is glucose 6-phosphate to fructose 1,6-bisphosphate. Here we have fructose 2,6-bisphosphate, AMP levels, and insulin also act as activator, whereas citrate, ATP levels, and glucagon act as inhibitor. So at the end of this first phase, which is the preparatory phase, you must have invested 2 ATP in step 1 and step 3. So after that, the glyceraldehyde 3-phosphate continues the journey. So it continues the journey here. So glyceraldehyde 3-phosphate will be converted to 1,3-bisphosphoglycerate in the presence of glyceraldehyde 3-phosphate dehydrogenase. Then 1,3-bisphosphoglycerate will be converted to 3-phosphoglycerate. And this particular one, ADP, is going to ATP, the phosphoglycerate kinase. And then 3-phosphoglycerate will also be converted to 2-phosphoglycerate. Okay, the phosphoglycerate mutase is the enzyme that catalyzes this. Why the phosphoglycerate kinase is the one that catalyzes the reaction between 1,3-bisphosphoglycerate to 3-phosphoglycerate, and the 1 molecule of ATP is being produced in this step. Okay, so you also have the conversion of 3-phosphoglycerate to 2-phosphoglycerate. Then you also have conversion of 2-phosphoglycerate to phosphoenol pyruvate. We have enolase as the enzyme, and we also have phosphoenol pyruvate to pyruvate. And the enzyme responsible for that is pyruvate kinase. And ATP is also being produced at this point. So this is the end of energy generation, or the payoff phase. That's the name of this particular phase. And so that marks the end of glycolysis. So what happens next to pyruvate determines which pathway is going to follow and the amount of energy that is going to go to. The pyruvate could go to lactate, the pyruvate could go to acetyl-CoA, and the pyruvate could also go to ethanol. So this is the general overview from glucose to glucose 6-phosphate to fructose 6-phosphate to fructose 1,6-bisphosphate, and then you have interconversion of dihydroxyacetone phosphate to glyceraldehyde 3-phosphate. Glyceraldehyde 3-phosphate continues the journey to 1,3-bisphosphoglycerate. 1,3-phosphoglycerate continues the journey to 3-phosphoglycerate to 2-phosphoglycerate to phosphoenol pyruvate, and eventually we have production of pyruvate itself. So the question now is, what happens next? So in the absence of oxygen, okay, in the absence of oxygen, for example, when you are exercising heavily, okay, when you are exercising heavily, and then it seems you don't have sufficient oxygen at that point, the pyruvate can be converted to lactate through a catalyzing enzyme called lactate dehydrogenase. Also, in some microorganisms such as yeast and some bacteria, we can have anaerobic respiration, that in the absence of oxygen, the pyruvate can be converted to ethanol. And the enzymes that are responsible for this are alcohol dehydrogenase and pyruvate decarboxylase. Okay, those are the two enzymes that can catalyze pyruvate conversion to ethanol in bacteria and yeast. Okay, however, in the presence of oxygen, the journey continues. So you have what we call the pyruvate dehydrogenase complex in the presence of NAD and the carbon 4 oxide. So the pyruvate is converted to acetyl-CoA. When it converts to acetyl-CoA, it moves to the Krebs cycle and then moves to electron transport chain. So, like I said, the absence of oxygen, no oxygen, is anaerobic respiration. And that is when the pyruvate can be used to make ATP, no matter how small. Okay. And at this point, the Krebs cycle cannot occur due to lack of oxygen. So, and the lactic acid in the muscle can cause muscle cramps. So by the time you have accumulation of lactate, okay, in the muscle cells, can lead to muscle cramp. Now, before we go on, it's important for us to note this phenomenon called the Warburg effect. Warburg effect. So, like we said, normally the glucose is supposed to be broken down to pyruvate, and the next thing is I should continue to acetyl-CoA, electron transport chain, or oxidative phosphorylation, where some ATP will be produced. However, you notice that sometimes in the presence of oxygen, the pyruvate will just not continue to form acetyl-CoA. But in the presence of lactate dehydrogenase, it will form lactate, and from there, it will produce so much ATP. To produce so much ATP times 100 to about from times, about 10 times to 100 times more, and you have the production of that ATP. That particular effect is referred to as the Warburg effect. So it's referred to as the Warburg effect. So I tried to demonstrate this here, where we have ATP produce M mass in the presence of oxygen. So these will actually empower cells. So this Warburg effect actually is like it empowers tumor cells. So you have tumor cells, you have a increased demand, you have increased demand of energy. So instead of the cell to go through all the processes, the cell goes through this Warburg effect, and it generates a whole lot of energy, and more cells will be produced. So you can see the energy coming up. The energy is in yellow, and ATP is in yellow. Okay, and you can see those balls that represent cells. You can see them proliferating, producing more, producing more, producing more, producing more. Okay, producing more through the effect of the Warburg effect. The oxygen, presence of oxygen in lactate that is being generated. Now, outside that, in the presence of oxygen, the reaction, the pyruvate is meant to continue the journey to acetyl-CoA, and this acetyl-CoA goes to link up with the Krebs cycle. So where we have the acetyl-CoA moving from isocitrate, then from isocitrate to oxoglutarate to succinyl-CoA to succinate to fumarate to malate and then to oxaloacetate. Okay. So like I said that the pyruvate is converted to acetyl-CoA. So here you can see a summary of the journey coming from glycolysis going to pyruvate, and then from pyruvate going to acetyl-CoA. Okay, and then because that acetyl-CoA combining to form the citrate, and the citrate to isocitrate. So this reaction releases 2 carbon 4 oxide. It reduces NAD to 2 NADH, and then produces 2 acetyl-CoA during this particular pathway. So this particular diagram shows the number of carbon that is present for each compound that is formed from acetyl-CoA, starting from 6 carbon compound to 5 carbon compound to 4 carbon compounds to 4 carbon compounds to 4 carbon compound, and eventually back to going to 6 carbon compounds. So it's an ongoing process, and it occurs in the matrix. That means the inner compartment. Okay, so the product that we get per glucose that has been broken down from glycolysis, that we have 2 ATPs will be produced at this step, 6 molecules of carbon 4 oxide will be produced at this step, 8 molecules of NADH, and 2 molecules of FADH2 will be produced in this particular step. So after this, these energy carriers now enter the electron transport chain. Okay, so you can see them. So in the electron transport chain, we have both the electron transport chain and the oxidative phosphorylation. The electron transport chain involves the transfer or the movement of electron from one electron carrier to the other, while the oxidative phosphorylation involves the process where ATP is formed as a result of the transfer of electrons from NADH to FADH to oxygen by a series of electron carriers. So the process in which ATP is being formed at the end is referred to that oxidative phosphorylation. Close that. So if you look at the stretch of the electron carriers, we have complex 1, we have complex 3, we have complex 4. Okay, and then we have the ATP synthase complex that helps in the production of ATP. Okay, so that region between complex 1, complex 3, and complex 4 is where the electron transport chain occurs. And this last region is the oxidative phosphorylation. Okay, so you can see we have said these complexes involve electron transport chain, while this space involve oxidative phosphorylation. So you have those complexes there. The NADH helps you carrying electron from food, donate electrons. So the protein complex of electron carrier is there. So the electron moves from complex 1 through its carrier to complex 3. And this leads to the building up of gradient on the intermembrane space. We have the inner mitochondrial membrane, then we have the mitochondrial matrix, that is the inner part of it. So we have the coenzyme Q that helps to carry electron from complex 1, that's NAD dehydrogenase to complex 3. Okay, and then we have another electron carrier, coenzyme C, that helps to carry electron from complex 3 to complex 4. Okay, so and at that point is when oxygen is also being utilized there to add to the hydrogen ion to form water. And thereafter, when there is a buildup of gradient on the intermembrane space, it leads to the ATP synthase acquiring enough energy to produce ATP from ADP. Okay, so you can see the ions, protons coming out through the complexes, just as the electron carries electron from one complex to the other. So you can see the buildup of the hydrogen plus the proton. So when you have the buildup there, so that buildup is what is going to lead to propel the ATP synthase to produce ATP, which is the energy of interest that we actually want in this particular case. So at the end of the day, of those three processes, the NADH from 2 or 3 ATP can be made. FAD 2 ATP can be made. So first glycolysis, we invested ATP, and then we get a net ATP of net energy of 2 ATPs and 2 NADH. And the Krebs cycle, we have 2 ATP being produced, 8 NADH, and 2 FADH2 has been produced. So overall, from glycolysis, we have 4 ATP, 10 NADH, and 2 FADH2. However, for the electron transport chain, we have massive production of ATP, about 30 ATP molecules being produced, 1 molecule of glucose. Okay, so when you add it to the 4 from glycolysis and Krebs cycle, so you have about 34 molecules of ATP that's been produced ab initio. So in writing it together, you can say we have a grand total of 38 ATPs that is being produced. So from the one produced from glycolysis, the one produced from Krebs cycle, and the one produced from electron transport chain. So we have a theoretical yield that's available. So all these yields, they are theoretical in nature. But in the real sense, we can actually have the production of this ATP that will be utilized in the body. So for every pair of electron transported to the electron transport chain by a molecule of NADH, between 2 and 3 ATP are generated. So for each pair of electrons transferred by FADH2, between 1 and 2 ATPs are generated. So for simplicity, however, we look at the theoretical maximum use of ATP per glucose molecule oxidized by oxidative phosphorylation. So let's take note. About 2 ATP from glycolysis, about 2 ATP from TCA cycle, about 30 to 34 ATPs from electron transport chain, vis-a-vis phosphorylation, the total will give about 38 ATP molecule. Okay, who is that now? Fanny Moku. Fanny Moku is not listening, just disturbing us. Now, do you have any question, please? Any question?
Yes, I want to ask a question.
Go ahead. Or Jelabi.
I could not hear what you were saying the other time about the ATP they produce in total. I saw something like calculation there, and I don't understand the process, sir.
Okay. So, I said for glycolysis, we can have at least, a theoretical extrapolation. So we can have 2 from ATP, okay? Then we can have 2 ATP from glycolysis, then 2 ATP from Krebs cycle, the total making 4 ATPs. Okay, so we have 10 NADH here and 2 FADH here. The NADH, they are reducing equivalent that is being used in the electron transport chain to carry electron across. And in carrying that electron across, it leads to generation of ATP, okay, in the electron transport chain. So both NADH and FADH2 carry out this role. So now, if NADH carries out this role, okay, and each NADH can generate 3 ATPs, say 2 to 3 ATPs, FADH can generate 2 ATPs, theoretically calculated, okay, and then you have it go 3 times in the electron transport chain. That is 10 times 3, because this 10 is from 8 NADH that was produced in the Krebs cycle and 2 NADH that was produced during glycolysis. 2 of them, 8 plus 2, making 10. So this 10 will be used during electron transport chain. So, and each one will produce 3. So 10 is going to be 10 molecule will be 3 times 10. That's 30. The FADH produces 2 ATP, okay, per molecule of glucose. So, and if you have 2 of them being produced during the Krebs cycle, so you can say the ATP they will produce, okay, is going to be 2 times 2, that is 4. So you are going to say 30 ATP plus 4 ATP, giving you a total of 34. So for electron transport chain, you have 34. So by the time you have the 4 from glycolysis, 2 from glycolysis, 2 from Krebs cycle, making 4 ATP plus 34 ATP. So the grand total will be 38 ATP. So the total amount of ATP will give you 38. It will give you 38. So if you look at this place, you can see FAD, okay, FADH2 and NADH2. So these are the ones that helps to donate the protons that enhances the movement of electron from one complex to the other. Okay, NADH and this NADH and FADH, they are being produced from the Krebs cycle here. You can see here FADH being produced here, NADH being produced here, NADH being produced here, NADH being produced here, NADH being produced here. Okay, so you can see it's FADH2, NADH, NADH, NADH. So they are being produced. So They have been produced, and some are also being produced from glycolysis. So this calculation is to give a theoretical outlook that, okay, take note that NADH produces about 3 ATP can be produced from it. FADH2, 2 ATP can be made from it. So under glycolysis, you have 2 ATP that's been produced and 2 NADH that's been produced. Under Krebs cycle, you have 2 ATP that has been produced and 8 NADH that has been produced and 2 FADH2 that has been produced. So in total, you have 4 ATP that has been produced between glycolysis and Krebs cycle. And then you have 10 NADH that have been produced between glycolysis and Krebs cycle, because 2 is produced from glycolysis and 8 is produced from Krebs cycle. And then FADH2 is only produced by the Krebs cycle and it's just 2. So there you have 4 ATP, 10 NADH, and 2 FADH2 between glycolysis and Krebs cycle. Now, if you now go to electron transport chain make uses of NADH and FADH2. So, and if you have 10 that is available, okay, and then it goes 3 times to produce the ATP. So per round is going to give you 3 ATP. So you can say, okay, 3 times 10, you can get 30 ATP from electron transport chain. The FADH2 can go 2 times, so you have FADH2 2 times 2 to give you 4 ATP. So the total will now give you 34 ATP. Okay, so I now said that before the generation of 34 ATP at the electron transport chain, glycolysis generates 2 ATP and Krebs cycle generates another 2 ATP on their own. So if you now add, and that makes it 4 ATPs. So if you now add 4 ATP plus 34 ATP, will give you a grand total of 38 ATPs in total, theoretically, that could be generated from glycolysis to oxidative phosphorylation through electron transport chain. Yes, any other question?
Thank you, sir.
Any other question?
Yes, sir. Good afternoon.
Afternoon, ma'am.
Actually, how many steps are involved in the Krebs cycle?
Can you speak loud, please?
Said, how many steps are involved in the Krebs cycle?
Okay. So, for the Krebs cycle, we have a 1, 2, 3, 4, 5, 6, 7, 8. Then 9th one goes back to citric acid. So the first one is when the acetyl-CoA binds with oxaloate acetate to give the citric acid or citrate. So you can count it here: 1, 2, 3, 4, 5, 6, 7, 8, 9. So, and this happens twice for each glucose molecule. The Krebs cycle happens twice.
Okay.
Yes, another question. So in the exam, of course, most of what I've mentioned is what is going to be considered. So all the explanation I've given is very, very important for you to take notes and read up on them. So I'll try to pass the notes across to Basit, so that Basit can get it across. Or have I sent it? Have I sent the note to you already?
No, sir.
No, sir, it's not clear.
No, sir.
Okay. Any other comments? Any other question?
No,
Sir,
So
I have a question, sir.
go ahead.
Sir, can we get the video, like the video recording of this class? Because there's a lot of things that you said that are not in the slides, sir.
Okay. Yes, you will get it. So maybe I'll post it on my YouTube. Most of you have not subscribed to my YouTube. I'll post it on my YouTube channel so that you can watch it there. What
Thank you,
about
sir.
that? Okay.
Thank you very much, sir. Thank you, sir.
Okay.
Sir, you will send the link, right?
Yes, you don't have the link. I
We
will
have the
send
link
it.
too.
Basit, is Basit there? Basit, are you in the class or is not there? It's not here.
Yes, I'm here.
You have the link now.
No. That's just the link to the class, not the link to the recorded class.
Okay, don't worry, I will send it to you.
Okay, sir.
Okay.
We also have botany students here. Sorry, sir. We
Go ahead,
also
please.
have botany students here.
You also have.
And the botany students.
Yes.
Yes, so I don't know who Basit is, so to get the slide also.
So how do we? I thought Basit is a contact link to all of you. We have botany, we have a integrated science education students, we have microbiology, we have marine, we have somebody should.
I will send the slide to the
Now
class
you
rep.
you should have a central. That's how we do it every year. We have a central class rep, probably from CBG. Who send to me. We'll contact all of that class rep. We send to your class members.
Okay, thank you, sir.
Well,
Thank you.
Okay. So in the absence of none, thank you very much. I wish you all the best in the exams. To score a very good score.
Thank you very much. Thank you, sir.
When are you coming to campus?
April, sir.
Oh, your own is April. Okay. All the best. See you then. Bye.
Thank you, sir.
Amen.
Thank you, sir.
Thank you, sir.
Come on.